All right, this is the second in our pathogenic bacteria series, and this one is going to look specifically at gram-negative Cocci and Bacilli. So let's talk a little bit and review what gram-negative bacteria are all about. They are the largest group of bacterial pathogens for humans, so much larger of a group than the gram positives that we looked at last time. They have something different about their cell wall. That's what we learned about the difference between gram-negative and gram-positive. They have a lipid type A in the bacterial cell wall, and that lipid A triggers fever, vasodilation, inflammation, shock, and disseminated intravascular coagulation, which is called DIC. So most gram-negative bacteria that are able to breach the skin, in other words, get into a place they're not supposed to be, or a mucous membrane, they like to grow at the same body temperature that we have, so 37 degrees Celsius, and they evade the immune system and are able to cause diseases in you and I. So here's just a little bit about that lipid A endotoxin that these gram negatives make, and just the different effects that they have and outcomes. So you can follow that flow chart and take a look at how some of these gram negatives are able to cause illness. All right, we'll start our discussion with looking at the gram-negative Cocci, specifically looking at Neisseria. So this is the only genus of gram-negative Cocci that regularly causes diseases in humans. These are aerobic, so they like oxygen. They're non-motile, so they don't have flagella or cilia or any of those things, and they tend to be arranged two together. So we call it diplococci. They are oxidase positive. When you do the oxidase test for them, they turn the little litmus paper. We'll be talking about this in lab. They turn the little litmus paper purple, and so you know that they are oxidase positive, and that is what distinguishes them from many of the other gram-negative pathogens that are out there. Pathogenic strains have fimbriae, which allows them to attach to things. They have a polysaccharide capsule, which protects them, and they have that lipid A that I just mentioned in the cell wall. There are two specific that are pathogenic to humans that we want to talk about, two species. One is Neisseria gonorrhea, and the other one is Neisseria meningitis. So here they're just showing you what the diplococci look like. So you have to look at those little guys there. You might want to blow up your screen so you can see what the diplococci, the two of them connected together, look like. All right, let's talk about Neisseria gonorrhea. So this is what causes the disease gonorrhea. This only occurs in humans, so this is not something you're going to see in any other animals. It is, of course, a sexually transmitted disease or sexually transmitted infection, as they often say now. And of course, you increase the risk of contracting this with increased sexual encounters. Most of the cases in the United States occur in adolescence, and cases have declined, thankfully, over the past few decades. It is more common in females than it is in males. So just taking a look at some charts here, you can see that the southern half of the United States, kind of wrapping up each coast, have a lot more cases of gonorrhea than other parts of the nation, but there's also a lot of population of people in some of those areas too. So you have to look at that. And then it shows a chart at the top that's looking at the incidence rates over years, and you see that we have experienced a dip in the number of cases of this. So the gonococci adhere to the genital, urinary, and digestive tract. They can evade the human immune system. They secrete protease that cleaves the secretory immunoglobulin IgA, and they survive within neutrophils. So when they're engulfed by a neutrophil, that white blood cell, then they are actually able to stay alive. So in men, they have inflammation that causes painful urination and pus-filled discharge. In women, they are often asymptomatic. And that is why they say you see more cases in women than you do men. Men are going to know about this right away. There's no doubt about this at all. The discharge is very obvious. In women, that's not always the case. And left untreated, it can trigger PID or pelvic inflammatory disease. Infections can also occur outside of the reproductive area, so different places in the body, in the mouth, in the throat, in the rectal region. You can also get an infection of the cornea or respiratory tract in a newborn when mom had gonorrhea during childbirth. This is diagnosed using genetic probes if it's an asymptomatic case. Again, the gram-negative diplococci in the pus that they are able to get a sample from, they can look at that in the lab and see that. So in females, it's a little bit harder for them to diagnose. The treatment is complicated because we have resistant strains. They use broad spectrum intramuscular cephalosporins for this. Prevention for this obviously is sexual abstinence, monogamy, and proper condom use. Eye infections in infants are prevented by using antimicrobials. So I'm not sure if they currently do this, but back when my daughter was born, a long time ago, they treated every baby, whether they thought they needed to or not. It was just a standard protocol to treat every baby just in case, because women can be asymptomatic and not know that. All right, let's talk about Neisseria meningitis. All right, this is the most common cause of meningitis in individuals under the age of 20. It can be normal microbiota that live in your upper respiratory tract. The bacteria are transmitted through coughing and sneezing or respiratory droplets among people in close contact. So this is an issue in dorms, in barracks, in heavily populated areas where people are living in very close contact with one another. Um, Meningitis can cause death within 6 hours of the symptoms, and meningococcal septicemia can also be life-threatening. One of the other things that the septicemia version of this causes is petechia, which is those red spots you see, that rash that you see, and then you can also see discoloration in the digits. This is diagnosed using using something that will rapidly diagnose it. That is a critical thing because, again, 6 hours. You only have 6 hours. So they use some testing for that. Gram-negative diplococci in the phagocytes in the central nervous system are evident when they do a spinal tap. The treatment for this is to administer antimicrobial drugs immediately. So they get you started on IV treatment of this right away. The prevention is a vaccine. So there is a vaccine that many of you, if you lived in a dorm, were required to get or heavily suggested that you get, so that you don't have to experience this. Asymptomatic carriers exist for this disease, and so it's going to be almost impossible to ever eradicate it. The best thing you can do is get a vaccination if you're going to be living in a dorm or in a barrack or anything like that. So Neisseria gonorrhea can be deposited on the vagina during sex, but can wind up infecting the uterine tubes in the pelvic cavity. Why is it able to do this despite being non-modal? Obviously, it's transported during sexual intercourse. All right, let's take a look at pathogenic gram-negative facultatively anaerobic bacilli. It's a lot of words. So we're changing shapes. So we're going from the cocci, which are round, to the bacilli, which are rod-shaped. And we're changing oxygen requirements. So when we were looking at the cocci, they were aerobic. These are anaerobics. All right. So there's two families that contain most of the human pathogens that we are interested in, the enterobacteria and the Pasteurella bacteria. The oxidase test is what's going to distinguish the difference between these two families. And there's also a lot of healthcare associated pathogens in this group. This is an oxidase test. Like I said, we're going to talk about it a little bit in lab as well, but it's basically you put the microbe on a piece of filter paper. And then so that's what they've done in the first picture there. They put the microbe on there. You can see it. And then you add a drop of a product called oxidase. And when you put that on there, if it's oxidase positive, it turns purple, like you see on the right-hand side. The left-hand side was oxidase negative. Okay, taking a look at this pie chart, it's just if you look at the little picture, it's the about equal amounts of gram positive and gram negative with a few yeast healthcare associated infections in the United States, so about half and half. And then when looking specifically at the gram negative bacteria, you can see that the majority of those are the enterobacteria. Specifically, Escherichia coli is the big offender there, but you can see that the other ones are in smaller amounts. So just giving you an idea of which bacteria are causing the most infections when we talk about nosocomial or healthcare associated infections. All right, so let's take a look at the enterobacteria. These are intestinal microbiota of most animals and humans. They are ubiquitous or everywhere in water, soil, and decaying vegetation. The enteric bacteria are the most common gram-negative pathogens in humans, as I mentioned before. These are motile, so they move, bacilli, and coccobacilli. So we haven't seen a coccobacilli. What that is it looks kind of like an oval, I guess would be the best way to describe that. It's not round like a cocci, and um, it's not rod shaped. It's kind of somewhere in between. These are all reduced nitrate to nitrite and ferment glucose. So these are biochemical tests, again, that we'll be talking about in lab. And they grow best in aerobic environments. The species are distinguished based on their biochemical properties, their motility, and their colony characteristics, so how they cluster together. All right, taking a look with a light microscope of the gram stains of the bacteria in this family. Dichotomous keys, we use these when we're trying to figure out what a unknown microbe is. And you do this through a series of, it's like a flow chart, but you have a series of two options for each um, particular characteristic. And so one of the things we'll be learning about in lab is all these different biochemical tests that you can run and what a positive and a negative looks like. So let's just look at this just for one little second here so you can kind of get the idea of how it works. So we start out with doing a lactose test. We've got an unknown bacteria. We don't know what it is. And so we're going to put a sample of it. We're going to inoculate the media that is used for this called rapid lactose fermentation media, right? And so we're going to inoculate that. And if we get a yellow color, then we know it's positive for rapid lactose fermentation. And if it turns red, we know it's negative. So let's just say for a second we determined that it turned yellow, and so it is a positive. The next test we would run would be a motility test. And so that motility test would allow us to determine if we have a positive or a negative, and we would go to the next step and then the next step. And so what will happen is at some point, like let's just say we're looking at motility and we determine that we have a negative on motility. We do a stab test for that. And we have a negative for that. We would follow the arrow and it would tell us that we have Klebsiella. All right, so that's how a dichotomous key for distinguishing bacteria is used in the lab. So this is a really common type thing that they do when we're trying to figure out what a microbe is. We're just going to run these series of tests and we're going to determine based on the outcome of each test. So a little bit more about the enterobacteria. The pathogenesis of this, there are a variety of antigenic components. The membrane components are variable. There are protein and polysaccharide capsular antigens. So remember, the capsule is outside of the cell wall. So there are both protein versions and polysaccharide and combinations of those. And we use that information to distinguish among the different strains and species, because we can run different tests and get positives and negatives. There are numerous virulence factors as well. Some are common to all of the individuals in that genre, and others are unique to certain strains. So again, we have tests that we can run to help us sort out what specific bacteria we have. So just a little bit of the factors that are associated, antigen and virulence factors that are associated with the typical enteric bacteria. All right, so they show you some of the antigens that we have out there. Common antigen, O antigen, lipid A that we talked about earlier. So those are some of the common ones so that you have their markers for. And then virulence factors, so things like fimbriae and endotoxin, adhesins, plasmids. All right, so all those different things that help to make it more virulent. So just a quick reminder what a virulence factor is. It's anything that allows a microbe to be a pathogen. So just as a little reminder, not all microbes are pathogens, all right? Only a small percentage of them are. But whatever these virulence factors are, different characteristics that allow them to be considered a pathogen. That's what a virulence factor is. All right, so taking a look at the diagnosis for this, we can diagnose enteric bacteria in urine, blood, and cerebrospinal fluid. There are biochemical tests that you can run in your lab that will help you to rapidly identify enteric bacteria. The treatment for this, well, most enteric bacteria, if it's digestive, in the digestive system, it causes diarrhea, and that is typically pretty self-limiting. However, if your patient is elderly or very young or has a compromised immune system, that might be a different situation. Internal infections are treated with antimicrobials. The prevention for this, of course, is good personal hygiene and proper sewage control. So we use an agar called McConkey's agar. We've talked about this a little bit in the lab already, that particular agar at least. And so you can see that they've streaked this particular plate. The agar itself is kind of like a pink color, a dark pink color, right? And so we've streaked the plate on one side with Escherichia coli or E. coli, and the other side with Staph aureus. And you can see that on that particular plate, that the first plate, that the E. coli grew rapidly, and you can see the discoloration or the coloring of the actual streak itself. The staph did not do anything on that particular plate. On the second plate, we're looking at E. coli, which is an enteric, and Salmonella enterica. And you can see that the Salmonella enterica grew and used up some of the coloring in the agar, and you got that yellow color. So when we look at the pathogenic enterobacteria, we are classifying them into three different groups. The coliforms, which rapidly ferment lactose. So you use a broth that has lactose in it, and it will ferment that. And when it ferments that, it turns the broth from a red color into a yellow color. The normal microbiota, these are normal microbiota, but they may become opportunistic in the right circumstance, such as somebody taking a long a long time they have to take antibiotics. There are the non-coliform opportunists. These do not ferment lactose. So when you put those in the media that has lactose in it, it stays red. And then there are true pathogens. So a little bit about coliform opportunistic enterobacteria. The coliforms are either aerobic or facultatively anaerobic. They are gram-negative, rod-shaped bacteria that ferment lactose to form a gas in the lactose broth. So not only does it change the color, but when you put a Durham tube, which is a little teeny tiny test tube, and you invert it and you put it in there, it has an air bubble in it. These are commonly found in soil and on plants and on decaying vegetation, and they colonize the intestinal tracts of humans as well as animals. Coliforms in water indicate impure water and poor sewage treatment. So let's talk a little bit about Escherichia coli. So this is the most common and important of the coliforms. E. coli antigens are used to identify the different strains. There's a bunch of strains of E. coli. Virulent strains have virulence plasmids. They have genes for the fimbriae, the adhesions, and the exotoxins. This can cause several different diseases. Gastroenteritis is the most common disease, which produces diarrhea and cramps and nausea and vomiting, but it's often mediated by enterotoxins. The major cause of pediatric diarrhea in developing countries is E. coli. So here's the bad boy, E. coli 0157:H7. This is the most prevalent pathogenic E. coli in developing countries. That means where we live. Causes diarrhea, hemorrhagic colitis, which is bleeding of the colon, and hemolytic uremic syndrome. All right, so this is associated with the consumption of undercooked ground beef or contaminated milk or fruit juice. It produces type 3 secretion system and Shigella-like toxin. All right, let's talk a little bit about Klebsiella. This is found normally in digestive and respiratory systems of both humans and animals. It has a capsule that protects the bacteria from being phagocytosed. It can cause opportunistic infections, and one in particular we want to talk about is specific here, Klebsiella pneumoniae. This is the most commonly isolated pathogenic species. It causes a form of pneumonia. It may be involved in bacteremia, meningitis, wound infections, and urinary tract infections. So this is Klebsiella underneath the microscope, and they used a special stain called a capsule stain. Capsule stain. And so you can really see that, right? So you see the bacteria, which is the dark purple little structure you see, and then the clear area all around it, that's the capsule. Serratia, this produces a red pigment when it's grown at room temperature. This can grow on catheters and saline solutions and other hospital supplies, and it can cause life-threatening opportunistic infections in someone who's immunocompromised. It's frequently resistant to antimicrobial drugs. You may have seen this growing in your shower. So if you get that kind of pinkish red growing stuff, usually in the corners of your shower and places like that, and it's a little slimy, that's almost always Serratia marsans. So it's red, right? So that's just what I mentioned. When you grow it out, it's very obvious. It's bright red. Some coliform opportunistic enterobacteria we want to talk about, Enterobacter, Hafnia, and Citrobacter. These are also found in soil, water, decaying vegetation, and in sewage. They reside in the digestive tract normally in humans and animals. Enterobacter can contaminate dairy products. All can be opportunistic infections. They can cause healthcare associated infections in someone who has a compromised immune system. They are difficult to treat because there are lots of resistance to them. So one in particular we talk about is Proteus. So it is a facultative anaerobe, and we'll talk about specifically Proteus mirabilis. And so this is most commonly associated with human disease, the one that Proteus mirabilis is. It's associated with urinary tract infections in patients that have long-term urinary catheters. And it's also can cause infection-induced kidney stones if you have this particular bacteria. It is resistant to many antimicrobial drugs. So this particular picture is the wave-like concentric rings. So you can see those of the swarming Proteus mirabilis. A few more to talk about: Morganella, Providencia, Edwardsiella. These are healthcare-associated infections that you see in immunocompromised patients. They primarily are involved in urinary tract infections, and then one in particular, Providencia, can cause kidney stones. The truly pathogenic enterobacteria, these are ones that you definitely don't want to come in contact with. This is Salmonella, Shigella, and Yersinia. These almost always are considered pathogenic due to the numerous virulence factors that they possess. They produce type 3 secretion systems. They introduce proteins into the host cells, and in doing that, they inhibit phagocytosis. They rearrange the cytoskeletons of eukaryotic cells, and they induce apoptosis, which is programmed cell death. Let's talk a little bit about Salmonella. This is a motile peritrichtius. That means the flagella are arranged all around the outside of the bacilli. They live in the intestines of birds, reptiles, and mammals. Most human infections are due to consuming food that has been contaminated with animal feces. Poultry and eggs are a common source of Salmonella, and they can cause salmonellosis and typhoid fever. So just a little picture of the events of this. So salmonella is ingested and it attaches to the epithelial cells that line the small intestine. Then it triggers endocytosis. The salmonella multiplies within the endocytic vesicle, and then the salmonella kills the host cell, inducing fever, cramps, and diarrhea. So a specific species of Salmonella is called typhoid fever. This is caused by Salmonella enterica, serotype typhi. Humans are the only host, and carriers are often asymptomatic. The bacteria is ingested in contaminated food or water, and the bacteria passes through the intestines into the bloodstream. The phagocytic cells ingest the bacteria and carry them to various organs. It causes gastroenteritis, bacteremia, and peritonitis, or infection in the peritoneum, which is the bag that holds all of your organs in your abdominal cavity. So looking at some incidence reports, this is looking out from 1935 to 2015. So a little bit older data, maybe 2016, right? And so it's showing you how few cases of typhoid fever we see anymore, and how many cases of salmonellosis we actually do see now. So you can see there's an interesting way that those two have kind of inverted. Salmonella is diagnosed based on the prevalence of the bacterium in clinical specimens. Salmonellosis is treated with fluid and electrolyte replacement. Typhoid fever is treated with antimicrobial drugs. The gallbladder has to be often removed from carriers to prevent infection because that's kind of where it harbors. Vaccines provide temporary protection to travelers in certain parts of the world, and prevention relies on proper sewage and water treatment, which is why we don't see as much of it as we could in places where we have pretty good water treatment. Shigella is oxidase negative, so remember the filter paper test, right? Non-motile pathogens, primarily a parasite of the digestive tract in humans, produces diarrhea-inducing enterotoxin, and there are 4 well-defined species of Shigella. There's Shigella dysenteriae, there's Flexneri, boydii, and Sonii. So Shigellosis is the disease that it produces. This is a severe form of dysentery. Sonii is prominent in the most predominant case in industrialized nations like the United States. Flexneri is predominant in developing countries. They're all associated with poor hygiene and sewage treatment. People ingest the bacteria present on their own contaminated hands or in contaminated food, and person-to-person spread is actually possible of Shigella. And then it shows you the events of Shigella, how Shigella actually goes into the cell and then ultimately makes its way to the bloodstream. It produces Shigella toxin, which is secreted by specifically by Shigella dysenteriae. It stops protein synthesis in the host cells and produces more severe disease. Shigellosis is typically self-limiting. It's treated with fluids and electrolyte replacement. Yersinia is a natural pathogen of animals. There's 3 important species of this. They all contain virulence plasmids, and they have adhesins. Again, adhesions always allow the microbe to attach to human cells. They produce type 3 secretion syndrome, which allows them to enables them to inject proteins that cause apoptosis of the macrophage and neutrophils in the host defense system. So some different strains of this that we want to look at. There is enterocolitica, which is acquired by the consumption of contaminated food or water, and it causes inflammation of the intestinal tract. There's pseudotuberculosis, which causes less severe inflammation of the intestines, and pestitis, which is highly virulent non-enteric pathogen that causes both bubonic and pneumonic plague. Okay, so if you remember a little bit, we talked a little bit about the Black Death back in the history of microbiology at the beginning of the semester, and we talked about the idea that the causative agent of the Black Death or bubonic plague is Yersinia pestis. Yersinia pestis is transmitted by fleas, and specifically they bite rats, and the rats have the bacteria, so now when they suck the blood of the rat, they now are the carrier of that particular pathogen, and then they bite the flea, not the rat, bites you, although the rat can bite you, but typically it's the flea that bites you, and that flea passes it on to you. So it produces in the lymph nodes, causes the lymph nodes to really swell, and they call that a bubo, which is where bubonic plague came from. So particularly evident in the groin area, the armpit area, and around the neck. So you have a lot of lymph nodes at all the portals of entry to your trunk, because all of the important stuff is in the trunk, right? Your heart, your lungs, all your intestinal organs are all in the trunk of your body, okay? And so you've got lymph nodes guarding all of those entrances. So your arms, your legs, and your neck from your head. All of the ways that pathogens can get into the trunk, you've got lymph nodes that are guarding those areas and protecting them. So this is showing you in the groin area, one of those lymph nodes that has swollen up. So diagnosis and treatment of Yersinia pestis must be rapid. Has a fast progression and it is extremely deadly. The characteristic symptoms are usually diagnostic, and there's lots of antibacterial drugs that are effective. So people think that the Black Death or bubonic plague is something that happened jajillion years ago, and that is true, but there are cases of it that still come up, particularly in the desert southwest of the United States, every year. So it's not ever really gone. It's just we don't see nearly as much of it, and we have an effective treatment for it now. All right, so this is just a chart showing you where the different bacteria that I've been talking about, which different systems that they might be found and create issues. All right, Pasturellice. These are small, non-motile facultative anaerobes. They require heme or cytochromes for growth. And there's 2 genera that we talk about that cause most of the diseases in humans, Pasturella and Haemophilus. So the first one that we want to talk about is a normal microbiota in oral and nasopharyngeal cavities of animals, so your mouth and your nose. Humans are infected via animal bites or inhalation of aerosols that contain the microbe. Most cases produce localized inflammation. You can have widespread infections and bacteremia that occur in immunosuppressed individuals, and diagnosis is by identification of the bacteria in patient specimens. There are antimicrobial drugs that are quite effective for treating this. All right, Haemophilus is a small pleomorphic bacteria. It requires heme and NAD for growth. It colonizes the mucous membranes of humans and some animals. Some species can become opportunistic, and Duckrelli is causes sexually transmitted diseases. So Haemophilus influenza is a pleomorphic that can assume many different shapes. That's what pleomorphic means. So all of these are the same bacteria on this slide, but notice that they're different shapes. So Haemophilus influenza, or H. influenza type B, HIB, is the most significant strain. It's a common cause of meningitis prior to vaccination. The HIB vaccine has eliminated most cases of Haemophilus influenza in the United States. Other Haemophilus influenza strains cause a variety of diseases, including conjunctivitis, sinusitis, dental abscesses, and meningitis. So a little bit to think about. Tell me why. Why is it that resistance to antimicrobial agents is more commonly seen in hospital-acquired infections with enterobacteria than in community-based infection with the same species? So think about the idea of you get it while you're in the hospital. So if you're in the hospital, your immune system's already probably not in the greatest shape, right? You're probably sick, possibly injured, but we're talking mostly about people who are there already because of some sort of illness. And so obviously their immune system is already suppressed somewhat because they're ill. And so we see more cases of it. Bartonella, this is an aerobic bacilli found in animals, but only causes disease in humans. So this is something that can be transmitted to humans from animals. There's 3 species that are pathogenic: Bartonella bacilliformis, and it causes Bartonellosis, which causes fever, anemia, headaches, muscle and joint pains, and chronic skin infections. This often can be fatal. It's transmitted by blood-sucking sand flies, and it's endemic in some countries in South America. Bartonella quintana causes a condition called trench fever. This was common during World War I. Transmitted by human body lice, causes headache, fever, and pain in the long bones, which are your legs and your arms. Causes 2 diseases in immunocompromised patients, including bacillary angiomatosis and bacillary pilosis hepatis. Sorry. Another one to talk about is Bartonella henselae. This is commonly known as cat scratch fever. It's transmitted through cat scratches and bites. A common disease in children in the United States, causes fever, malaise, and swelling at the infection site. Bartonella infections are treated with various antimicrobials. And there's what cat scratch disease can look like. So Brucella is a small non-motile aerobic coccobacillus. It can infect both humans and animals. Brucella mellitinus is responsible for all diseases in humans. It causes brucellosis. It's often an asymptomatic or mild disease. Human infection is typically through contact with contaminated dairy products or infected animal parts, and it produces a fluctuating fever. So you have a fever, it goes away, you have a fever, it goes away. Treated with various antimicrobial drugs, and there is a vaccine that's used in domesticated animals for this. A chart looking at the incidence of this disease in humans in the United States from 1971 to 2014. So obviously on the decline. Let's talk a little bit about Bordetella. This is a small aerobic non-motile coccobacillus. Bordetella pertussis is the most common. It is the causative agent of whooping cough or pertussis. Most cases of this disease are in children. The microbe produces adhesions and toxins that mediate the disease. They're listing the toxins there for you. The bacteria is inhaled in aerosols, coughing and sneezing, and it multiplies in epithelial cells. So, reported cases of pertussis in the United States from 1960 to 2014. Notice that in 1960 there were a lot of cases of it. And then they had a vaccination. And once that vaccination came around, you notice the number of cases went down dramatically. But you also see that cases started to go back up because of people's choosing to not vaccinate. Here is Bordetella, a scanning electron micrograph of it. The Bordetella are the pretty pink things. This is a timeline for the progression of pertussis, so it's just showing you the different periods, incubation, that whole idea of the different periods of time, and then the symptoms you would expect to see in each of those. So the diagnosis of pertussis is usually diagnostic. They have a cough that has a very characteristic whoop at the end. So they'll cough and they have this sounds like a whooping crane if you've ever heard one. That sound at the very end when they're coughing. The treatment is primarily supportive. There is an immunization for this, the Tdap. The Tdap is diphtheria, tetanus, attenuated pertussis. All right, Burkholderia. Um, the first one we're going to talk about here is Burkholderia cepacia. This is an aerobic flagellated beta proteobacterium that can decompose a broad range of organic molecules. It is used in the cleanup of contaminated environmental sites, but is also used by farmers to reduce fungal infection in plant crops. It can, unfortunately, become an opportunistic infection or pathogen in a cystic fibrosis patient, and it is resistant to many antimicrobial drugs. Another strain of this is Pseudomallei, which causes melioidosis. The pseudomonads, these are aerobic bacilli. They are ubiquitous in soil, decaying organic matter, and moist environments. These are problematic in hospitals because they are opportunistic pathogens. So Pseudomonas aeruginosa, this is a microbe that's rarely found as a normal human microbiota. So this is not something that typically lives inside of your body. It rarely causes disease, despite having a lot of virulence factors, including fimbriae, adhesins, capsules, toxins, and enzymes. However, it can cause opportunistic infections in immunocompromised patients, and it can colonize almost any organ or system in the body. It also infects the lungs of cystic fibrosis patients. It produces a biofilm, which protects the bacteria from phagocytosis, and the treatment for this is difficult because of multiple drug resistance. So a patient with Pseudomonas aeruginosa infection. All right, the Pseudomonads, Moraxia, and Acinetobacter. These are aerobic short plump bacilli that rarely cause disease in humans. There are a few that we want to talk about. Moraxilla catarrhalis, which can cause opportunistic infections in the sinuses, bronchi, ears, and lungs. And Acinetobacter, which grows in the soil, water, and sewage. It is an opportunistic infection in the respiratory, urinary, and central nervous system. Francelia tulinarius, which is a non-motile, strictly aerobic Cocci bacillus. It is an intracellular parasite of animals and amoebae in the water. There is a diverse range of hosts. Reservoirs in the United States include rabbits, muskrats, and ticks. Tumularia is a zoonotic disease. So zoonotic means that you can get it from an animal. It's spread through the bite of an infected tick or contact with an infected animal. It also can spread in meat, water, and aerosols. The bacteria is highly infectious. It can be misdiagnosed. The symptoms are similar to several other diseases. It's treated with antimicrobial drugs, and infection is prevented by avoiding reservoirs of Francisella. Legionella. This is an aerobic, slender, pleomorphic bacteria. It is a universal inhabitant of water, so it's always found in water. Humans inhale the bacteria in an aerosol from water sources. This becomes an intracellular parasite, and Legionella pneumophilia is what causes most diseases in humans. So here it's showing you an agar that we haven't talked about. Buffered charcoal yeast extract agar. And so that is, if you suspect Legionella pneumoniae, then this is the agar you would use for that. Remember, I said there's about 50 different agars out there, and some of them are used a lot, and some of them are used very rarely, and this is definitely one you would not use very often. All right, Legionnaire's disease. This results in a form of pneumonia that can be fatal, especially in someone who is immunocompromised, or Pontiac fever, which is similar to Legionnaire's disease, but it doesn't produce fatal pneumonia, and it's typically not fatal. The diagnosis for Legionella is using an antibody staining or other serological test in a med lab. The treatment for Pontiac fever is that it's self-limiting, so you just let it run its course. There are a couple of drugs that we use to treat Legionnaire's disease. The prevention of this, well, it's hard to prevent because the elimination of the bacteria is not feasible. So where they have the most problems with this is in HVAC systems. And so in older HVAC systems, like a lot older HVAC systems, is where this was an issue for a while. But mostly they've upgraded all of the HVAC systems and they don't work the same way, and it keeps the bacteria from growing in the system. Coxiella burnetii. This is an extremely small aerobic bacteria. It's an obligate intracellular parasite, so it has to get inside of a healthy cell. Originally, it was thought to be a virus because that is what viruses do as well. Its infective body enables it to survive in harsh environments. This causes human diseases that are associated with farm animals and pets. It's transmitted mainly by inhalation of infective bodies. So there's Coxiella. Again, intracellular parasite. So this is a scanning electron micrograph, and you see the little blue things, and that's inside of a bacterial cell. Q fever. This causes fever, headache, chills, muscle pains, and mild pneumonia. Chronic Q fever can cause endocarditis years after the initial infection. Chronic Q fever is treated with long-term antimicrobials. There is a vaccine that has been developed but is not readily available in the United States, and this is of course prevented by avoiding the inhalation of contaminated dust. Why do most U.S. cases of tularemia occur in the late spring and summer months, while fewer cases occur in January? So I want you to go back and look at that tularemia. Couldn't say that a second ago. Where that occurs and why that occurs, and think about the weather in the spring and the summer compared to the winter. All right, some pathogenic gram-negative anaerobic bacilli. These are mostly found in the microbiota of your gastrointestinal, urinary, reproductive, and lower respiratory tracts. These are actually important for human health. They inhibit the growth of most of your other pathogens. They synthesize vitamins and vitamin precursors, and they aid in the digestion of food. But if they get into a part of the body where they're not supposed to be, then they can cause disease. The bacteroids, these are normal microbiota of the intestinal and upper respiratory tract. Flagellus is the most important pathogen of this genus. It produces various virulence factors, which allow it to cause disease. It's involved in a variety of conditions, including abdominal infections, genital infections in women, and wound infections on the skin. And then there is a treatment for this. All right, so here is Flagellus looking at a plate. So you see that it's been streaked on there and it's caused some discoloration. Prevotella. This is a normal microbiota that you find in the urinary, genital, and upper respiratory tracts. This is different from the bacteroids in their sensitivity to bile. It produces several different virulence factors. It's involved in various conditions, including sinus and ear infections, almost all periodontal, which has something to do with your teeth infections, gynecological infections, brain abscesses, and abnormal infections. It's treated by surgical removal of the infective tissue and then some medication. So they ask you, why are Prevotella and bacterioids unlikely to opportunistically infect skin? So think about where those are located. And that's it for this particular chapter. So again, I would make myself maybe a spreadsheet for this and look at your different pieces of information, the diseases caused by each, the treatments for them, all those kinds of things. And I will see you for the next lecture.